Hi, I'm Will Ederer, a researcher in the FURI program at ASU under Dr. Yuqiang Bi. The FURI program stands for the Fulton Undergraduate Research Initiative, and under this initiative I investigated a project which I called Facilitated Transport of Contaminants by Microplastics in Porous Soil Media. That's a big mouthful. Basically, we investigated how microplastics, which are very small pieces of plastic, could move around in soil. We also looked at how contaminants, such as harmful environmental toxins, could stick to the plastics and potentially move around with them, which is what the facilitated transport part means. To study all these things, we decided to run experiments in a soil column. We used glass beads as a stand-in for real soil, and we pumped water with trace amounts of contaminants through the columns and then measured the concentration of contaminants coming out. We added microplastics to some of the columns and compared them to columns without microplastics to see if plastics had an effect on the concentration of contaminants leaving. We could also observe how microplastics moved through the column by looking for microplastics in different sections. We started with all our microplastics in the top, and then checked the rest of the column for evidence of them at the end of the experiment. To help with viewing the plastics, we dyed them with Nile red, which makes them glow red under certain colors of light. We decided to use two contaminants. The first was lead chloride, which can occur in soil from various contamination sources, and atrazine, which is an herbicide that can be found in agricultural soils. The contaminant solutions were dissolved in water at a concentration of 1 part per million, or ppm. Overall, we tested 6 columns, 3 for each contaminant, one of which was a control, one had polyethylene microplastics, and the other had polypropylene microplastics. To summarize, we took 2 contaminants, lead and atrazine, dissolved them in water, and then pumped them through a column filled with glass beads and topped with microplastics. Then, we measured the concentration of the contaminants in the water that left the column, the effluent, and scooped out different sections of the column to look for microplastics through a fluorescent microscope. We measured the concentration of the contaminant in the water that left the column over time. This graph shows the atrazine concentration. The effluent measurements are the data points, while the incoming concentration is graphed as a dashed line. Overall, the measured data stays pretty close to the incoming concentration, so we can see that a breakthrough of contamination occurred early and that the glass beads and plastics did not do much to absorb the atrazine from solution. The next graph shows the lead concentration. This time, the data points are on a different scale than the incoming concentration. The data points are in parts per billion rather than parts per million, because there are a few orders of magnitude smaller than the incoming concentration. This tells us that the glass beads and column absorbed lots of lead, because even the data in the control shows very low concentration. Additionally, the concentration of lead leaving never significantly increases, signaling that a breakthrough did not occur within the 48 hours of the experiment. The difference in the effluent data for the control and plastic experiments could be due to the presence of plastics, but the difference is fairly small compared to how much was taken out by the glass beads. Once the experiment finished, we scooped out sections of the column and removed plastic particles to look for movement through the column. Looking at the microplastics under a fluorescent microscope yielded these pictures. As can be seen, there were many plastics that just stayed in the top. However, a noticeable amount traveled to the middle of the column, and the ones that did tended to be a little smaller than those that stayed in the top. Fewer particles made it all the way to the bottom, and these tended to be smaller than the particles that made it to the middle. This shows that microplastics could travel the entire length of the column, and that there was a little bit of size separation going down the column. We saw that overall, atrazine did not seem to be well absorbed by column components, while lead did, and that the difference between the measured concentration in the control and plastic experiments could have been due to the presence of microplastics. Also, plastics were able to fully travel down the length of the column. This points us to the possibility that microplastics could adsorb contaminants and facilitate their transport through soil during infiltration. Future research could focus on a smaller size range of microplastics to see how many can travel down the column, and could look at adsorption capacity of different materials. Thank you for watching. If you want to check out my poster, you can find it at furi.engineering.asu.edu/symposium.